for the construction of 16 plasmid-based gene libraries, which were generated by error-prone PCRs. These libraries can now be used in directed evolution experiments to isolate highly active A-team variants with the ability to accommodate and isomerize longer sugar substrates than those of wild-type TIM. Parallel to these biochemical experiments, a process for the efficient production of recombinant enzymes developed in the group of Professor Neubauer was adapted to TIM by Mr. Castellan. Although the structure and function of TIM has been investigated since decades, some aspects of the reaction mechanisms have remained enigmatic. For example, the coordinated motion of the active site loop 6 and 7 upon substrate binding has been shown to be essential for the positioning of the neighboring catalytic glutamate 167. However, it was unknown how the N-terminal and C-terminal hinge residues of loop 6, proline 168 and alanine 178 contribute to this motion. In the second part of the thesis, Mr. Castellan has generated the P160A and A170L mutants by TIM by site-directed mutagenesis and characterized the purified proteins by steady-state enzyme kinetics and X-ray crystallography in the presence of the transition state analog 2-phosphoglycerate. The results show that proline 168 is important for catalysis because the plane of its side chain is crucial to transmit a conformational transition to loop 6, which is initiated by the binding of the substrate phosphate to loop 7. Residue alanine 178 seems to be less important for catalysis. Also, loop 6 is an